morning, everyone, and thank you, Professor Estacio. My talk for today will be on the production of STEM, science, technology, and engineering and mathematics PhD graduates learning from the first decade of the AST-HRDP and ERDT programs. The first decade of the Advanced Science and Technology Human Resource Development Program and the Engineering Research Development Program, Development for Technology Program, is a good source of lesson for us to be learned in the production of STEM PhD graduates. As a background, the National Institute of Physics offered the first PhD in physics in the early 1980s. And at that time, the Department of Science and Technology had a program called the UP Ateneo de Manila University de La Salle University PhD Consortium. And the first batch and second batch of scholars were the first batch was 6 and the second was 6. So the magnitude and the number of scholars that were awarded between the AST-HRDP and the UP Ateneo de La Salle PhD Consortium was one order of magnitude in difference. There is a need for us, and everybody agrees, including the government, to increase the number of scientists and researchers in this country. And normally, the metric that we are looking at is the number of researchers, full-time equivalent per million of population. But in this slide, I would like to point out that we have a problem with the figures. If you look at the left-hand side, in 2013, the number of researchers per million in the Philippines was 187. Then it jumped to 2015 to 268, and then 321, 385, and 418. If we look at these figures, this country has no more problem satisfying the UNESCO prescription of 380 researchers per million of population. So, and this is not true, obviously. And you can see that from 2002 to 2011, the average number of researchers per million of population was 78.8, almost 79. And then it suddenly increased to 187 in 2013, an increase of 138%. And during this period, there was nothing that really happened with regards to the training of scientists and researchers. So I chose not to look into those figures for certain sampling issues. Now, this is the S&T human resource feeder structure that we have in this country. We have the high school, and in academic year 2019-2020, there were 3.125 million senior high school students. And in terms of specific source of good STEM undergraduate students, we have the Philippine Science High School that is operated by a DOST. It has a national presence with 16 campuses. In its total enrollment in 2018-2019 was 8,358. So if we divide that by 6 levels, we have around 1,200 graduates a year from Philippine Science High School alone. And there are other 53 science high schools and several high schools, private high schools with good STEM high school training programs. If we go then from high school, they go to tertiary education, and you can look at the STEM enrollment, which is 411,831 in 2017-2018. And then we go to graduate school. And we can see that the number of students decreases almost by one order of magnitude as we go from high school, tertiary, to graduate school. And there are now 10 universities in the country that are offering PhD degree programs in STEM, and 8 with PhD programs in the engineering sciences. If you recall, in the 1980s, there was only one university with a PhD in physics in particular, and this was UP. And people have to pull all the necessary faculty resources from Ateneo and De La Salle included in order to have a better roster of teachers and faculty. The Philippine higher education system in 2019-2020 consisted of 1,975 universities and colleges, 87.5% of them privately owned. There is an increase. If you look at in 2018-2019, the number was 1,963, or an increase of 12. And the year before that, a large increase of more than 50. And there are 3.41 million undergraduate students, 46% of them studying in public institutions. And there are more than 230 of them across the country. Now, enrollment in STEM is a small portion, below 20%. And in the sciences and mathematics is even lower at below 2%. The biggest challenge that we have in the training of STEM PhD graduate is the lack of qualified PhD faculty with the requisite PhD degrees. Across the entire higher education system in 2003-2004, only about 10% of the entire faculty members had PhD degrees across all disciplines, including the humanities and the social sciences. It increased to 16.58% 2 academic years ago in 2018-2019, and then decreased again to 15% in 2018-2019. Probably it is due to the rosters of the newly accredited schools where the ratio of the PhD to MS was not favorable to increase the percentage. So the increase in the number of PhD faculty year to year is a lethargic 0.38% per year. So there is a need to produce more PhD graduates, if only to improve the PhD faculty profile of our higher education system. This is the budget for the SUCs has increased, and also the 3 times relative to 2006. And for the DOST, it also increased 6 times. If you look at the National Expenditure Program per year, it is about 20% of the gross domestic product in the previous year. So it is important for this country to have its gross domestic product growing so that its national budget is capable of growing without too much borrowing that the future generations will have to pay. And among there are 112 SUCs that are operated using a portion of the national budget. Out of this budget from 2006 to 2016, 27% or more than 1 in every 4 pesos goes to UP. So because UP is the national university, but UP is just one of the 112 SUCs. So looking at this, I always argue that UP has a disproportionate responsibility to solve the lack of PhD graduates in this country because it is given more money. Now, this is the faculty profile of the accredited higher education institutions in the country. And in 2003-2004 academic year, more than 60% of faculty members had bachelor degrees. Then it decreased, and in 2017-2018, 40% of faculty members had MS degrees. But still, these faculty members are not qualified to supervise PhD students. Now, again, the growth rate in the number of PhD faculty in the percentage is only 0.36% from 2003 to 2020. As a result, 99% of our higher education institutions are not capable of offering tenable STEM PhD degree program, even one program, because of the lack of qualified PhD faculty members to teach and to supervise graduate courses. Now, the same higher education system produces 1,538 new lawyers a year, and passing the bar is one of the most difficult professional exams. Only 1 out of 4, basically, only one out of 4 bar takers are able to pass and become member of the bar. The number of new certified accountants per year is almost 2,000 at 1,924, passing rate 30%. The number of new medical doctors we produce is almost 3,000 per year. The number of STEM PhD graduates, there is no official figure from CHED or anywhere. And my estimate is 100 PhD graduates in STEM. Now, an issue with regards to the retention of talented high school students to the STEM undergraduate degree programs, particularly UP, is the nature of the UP college admission test. The number of UPCAT applicants is increasing. As you can see, in 2003, you have 64,000. 56.2% of them would like to enter UP Diliman. And at that time, the number of slots for UP Diliman was 3,820. Come the year 2020, meaning to say UPCAT in 2009, the number of applicants was 92,000, and 65.6% of them would like to get into UP Diliman. But the number of available UP Diliman slots has stayed basically the same at 3,920. And if you compare that in 2003, it's just an increase of 100. But the number of UPCAT takers from 2003 to 2019 increased by 30%. So you can just imagine how many talented students are not able to enter UP, particularly UP Diliman. Okay, so that is a big challenge for this university. Now, the engineering research and development for technology program was initiated in 2007 with 2 aims: to support graduate scholars, particularly in MS and PhD degrees in engineering, as well as provide equipment and research grants to UP faculty. There were 8 partner institutions when it started, and half of them were public institutions, and the other half were private institutions. And 3 are based in Metro Manila, and 5 are in Luzon. Of direct concern to us in physics is the Advanced Science and Technology Human Resource Development Program. started, or at least planned in 2007. At the start, there were 9 institutions, and so meaning to say, these were the institution with uh, PhD programs at the time. Out of the 1,900, only 9 institutions qualified according to the DOST Science Education Institute. Then later on, the University of San Carlos joined in, where to become 10. So now, this is a list of scholarship grants granted under those programs. You can see that in 2008, there were 302, and in 2018, it became 1,270 PhD slots. And the target in 2019 was 1,985, and then to 1,927 in 2020. Now, the scholarships that is provided by the DOST through the AST, HRDP, and ARDT programs are generous, as listed here. They allow a scholar to study full-time and spend full-time of the research in the laboratory. The 2 programs are managed by the Steering Council, that is composed of a representative from the DOST, particularly the SEI director, and the deans of the participating partner schools. For the case of UP Diliman, it is the dean of the College of Science, the incumbent dean of the College of Science, and the dean of the College of Engineering as representative. It is important to understand that the DOST does not grant PhD degrees. Only the schools that are participating. And this is the AST HRDP performance from 2007-2018, and PhD scholars in 2007-2015. Shown here is AST HRDP program, 662 were awarded, but only 373 of them graduated. That's a 56.34% success rate. But notice that there are a number of institutions that perform better than the average, particularly the Visayas State University, 100% graduation rate. And we have to remember, AST, HRDP, and ARDP is publicly funded. This is part of our taxes being paid every year. Now, notice that the 3 participating UP institutions perform below average. UP College of Science, only 34% of its 99 PhD scholars were able to finish. And notice that we counted the scholars until 2015 and counted the number of graduates until 2018, because the PhD degree is programmed to be 3 years after MS. ERDT performance, only 48% of the PhD scholars that were awarded within the same period, only 129 out of 268 graduated. And the UP College of Engineering performed better than the average of the ERDT program. So we have a problem because the success rate is hovering at 50%. Now, let us look at the performance of the College of Science and the College of Engineering in terms of PhD graduate production from 1990 to 1991 to 2018-2019. And you can see that for the College of Science, there is no trend towards an increasing number of graduates. In fact, there are periods where there is an increasing trend, 5-year period basically, but it is not sustained. From 1990 to 1995, the number increased, but then it went down to the bottom until 2002, 2003, and then increased again. So what does that tell us? There is no effective program or strategy. No? The College of Engineering, on the other hand, was performing badly from 1990 to basically 2010. But then with the introduction of the ARDT program, the number of PhD graduates increased. So the college benefited from the ARDT program. And you can see here that from 2010 to 2011 to 2018, 2019, in fact, the College of Engineering, with its smaller faculty number, is producing more than the College of Science in terms of PhD graduates, 13 versus 12.24. Okay? Now, UP Diliman, if you notice here, increased its PhD faculty from 498 in December 2015 to 607 to February 2020. That's around 30% increase. Also, the College of Science to 168, but then the number of PhD graduates remained the same. Okay, wishful thinking. There must be one PhD graduate for PhD faculty every 3 years, and if it is achieved, the College of Science at the present faculty complement will produce 55 PhD graduates per year. College of Engineering will be 25. The College of Engineering at this point is already at 40% of that desirable value. Recommendations. The steering councils of this program need to do more to improve program performance. There should be more Filipino mentors to train PhD students to become successful independent researchers. And one aspect that should be looked at is to allow Professor Emeriti to supervise PhD students. All those who are good, and create a nurturing and enabling environment, and make successful mentoring of a PhD student a requirement in the grant of tenure and full professor appointment. Last recommendation, prior acceptance of PhD dissertation research findings and peer-reviewed journal, which is now done by the National Institute, which has been done by the National Institute of Physics for a long time. Make successful PhD mentoring matter most in SNT and recognition system. Increase the number of STEM undergraduate admission so that the RA 10931 is realized, and start getting free access to STEM PhD and MS degree programs. Thank you very much for your kind attention.
Okay. Thank you very much. Okay. Yes. My research is development of microscope, especially for biological research, and so not only for the biology, also medicine is on the target. And the research in the medical or biological research is of course use a lot of optical microscope quite often. And one of the reasons why optical microscope is so useful is it's a non-invasive technique to image samples, especially on live cells, live tissues, and it provides 3D resolution, and it allows observation sample in water, which means so the cell can keep kept physiological conditions. And it can, of course, do material detection, like using a spectroscopy, and we don't have to touch cell. So remotely, it can be modified. And also there are many other techniques like fluorescent probes, which highlight your target molecules. So those powerful tools are actually supported by, of course, the optical imaging technologies. And especially the optical imaging technologies has a lot of attractive parts. From the point of the physics, it provides a lot of different imaging mode. So these are just 4 examples. If you use different optical effect, you have different contrast. That means you have different information in the sample. So So like fluorescence, you have to stain sample by fluorescence molecule, but the staining technique, you can highlight your target molecule, target proteins, or even the organelles. And the dyes molecule, which is used for bright field imaging, it can separate cytosol or nucleus or cellular structures. And of course, you can use the other information molecule or on the sample itself, like refractive index or scattering efficiency. Those can give different contrast. And today, I'd like to introduce a new modality recently developing, which is Raman scattering. And the Raman scattering is known as a powerful technique for spectroscopy, and it actually detects the molecular vibrations by measuring by its spectrum after scattering. And this energy diagram simply shows how this Raman scattering monitor the molecular vibrations. And the instant light is given to the molecule, and after interacting the light, the molecule, it excites the molecular vibrational state. And since this vibrational state excitation requires a portion of energy of light, the other energy of light is scattered as a Raman scattering light. So if you measure the spectrum of scattered light, you can tell how much energy is taken to excite the molecular vibration, which is actually related to structure of a molecule. And this is an example of Raman spectrum we can measure from the side of a HeLa cell. It's a human cancer cell. And if you shine 532 nanometer wavelengths green laser to cell, and then you detect the spectrum of scattered light from the 550 to 640. It's a green to red region, and you can find many peaks. And these peaks are Raman scattering. And we can assign these peaks to different molecular species or molecular structures, and which is usually shown as a Raman shift, which is proportional to the energy of the excited to the vibration modes. And basically, we can use those informations to analyze the cell itself or intracellular structures. And in our study, we develop a microscopic technique, the imaging technique using this Raman scattering. And in Raman imaging, we detect Raman spectra from every defined point of the view area. So this is a phase contrast image of a cell. And after measuring Raman spectra, we have a data set, X, Y, and the spectrum. So this cubic data set, it's a huge amount of data. However, it contains lots of information of molecule in the cell. And by picking up molecular vibrations or Raman peak of our interest, we can map the distribution of the Raman scattering, and which shows the different molecular species in the cells. These three are typical image we can easily obtain from living cell. And the 750, this can be assigned to pyro ring breathing mode of cytochrome in the protein, which is seen in the mitochondria. So we can see the contrast of mitochondria clearly from this vibration mode. And 1683 is peptide bond vibrations called amino 1 mode. And this is seen in the protein beta sheet. So we see a lot of different, we see the signal from entire cell body. And lipid CH2 stretching mode. So this is seen in a lipid droplet and the organelle membrane structures. And we can roughly separate molecular species by using the vibrational molecular vibrations, and they can make a contrast without labeling. So this Raman scattering is really very it has a long history as a spectroscopy technique. However, it's not been used in microscopy for a long time. So the reason is the signal weakness of the signal. And if you compare the cross section of gamma scattering and the cross section of absorption of the fluorescence molecule, you can tell how weak this gamma scattering signals is. So basically, it takes a long time to measure gamma spectrum, and then it makes difficult to image live cells. So this is some example. So with using a model laser power, and with a few to several seconds of exposure time per 1 point. And if you scan this 1 point illumination and scanning sample, it takes about 20 hours for 128 pixels. So it's too long for live cell imaging. So in our system, we developed illumination optics that can detect gamma scattering, gamma spectra from more than 100 points at the same time. So by using this, we can shorten the imaging time to about 10 or 20 minutes. Then we can image the live cells in a reasonable time. So it's not very fast, but still the cell can stay there while we are observing. So the slow dynamics of cell can also be obtained. And so I just skip this one. And this is the example of imaging. And in this case, we used 120 lines of illumination and 5 second exposure time, and they can acquire the cells in 15 minutes. So it's not very fast, but still cell remains in the same position, and we can see the distribution molecules. And after developing this system, we did many applications like the protein signaling apoptosis and the differentiation of the breast cells, or like the things that molecular dynamics in cell divisions. And the Raman microscope is a new modality, so it gives offers label-free imaging and also analytical imaging. So we have a spectrum of cells. You can use spectrum to identify cells like cancer cells, non-cancer cells, or differentiated or non-differentiated cells. So those are new applications of the microscope. And in addition, we recently proposed Raman microscopy as a new modality for small molecule imaging. So what's a small molecule? The small molecules are just small. So that's about the size. So it's a size. The size matters because we basically cannot see it in the living cells because we cannot label it. So usually to image molecules of interest, and specifically we have to label it like fluorescent molecule. However, these molecules are too small to be labeled. If you label them by a fluorescent molecule, it changes chemical structures a lot. So what you are seeing is different from the natural behavior of the molecule. Of course, Raman scattering can detect it. However, since there are many different kinds of molecules existing in a cell, we cannot separate only your target molecule in this crowded fingerprint lesions. So we instead try to use this silent region, which is it's not crowded, so we can separate big, small easily. However, this also means no particular peak of your target molecule. So we try to use this alkyne, which has the Raman peak at this silent region, about 2100, 2020. And by using this alkyne as a tag for small molecule, we can separately detect your target molecule. So this is a collaboration work with Professor Sodaka and Dr. Dodo in WeChem. They are sensitive chemistry group, and we worked together to use this alkyne as a tag for molecule. So this is one example, EDU. So the oxy origin is labeled by this alkyne. And if you take major Raman spectrum, this alkyne, you see this strong alkyne peak in the silent lesion. And of course, this is different from the original origin, and it can be separated from the cell spectrum. And for the proof of concept, we incubated cells with EDU, and the origin have a similar structure to thymine. So when the cells synthesize DNA, it is incorporated into the nucleus. And we observed this cell by round microscope after incubation of 6 hours, and we clearly observed this alkyne signal located in the nucleus, which means the EDU was taken into the nucleus without losing their functions. And no EDU, of course, no signal from the cell. So this is the first demonstration of using EDU as a small tag, alkyne as a small tag for a molecule. And we also developed different kinds of tags, and this one, ubiquinone alkyl has a 2 alkyne, and this has different Raman peak as shown here. So we can perform the multicolor alkyne level imaging. And we also developed different tags for lipids, and also this chemicals, which has the nitrogen, which also has a peak in the cell region. And the organ imaging, we can also use this Raman tag techniques. And this technique now expanding in other groups, and so we can apply this to many different molecules, from the drug to protein lipid molecules, glucose. So it's many possibilities. However, the current detectable limit is about 200 micromolar. So that means we need high concentration of the molecule. So that actually limits the applications to only the target, which can accumulate in a small area of the cells. So now we are trying to improve this sensitivity by using many different methods. And sorry, how many minutes I have? So it's also about the time, I think.
Yes, pretty much.
Yeah. Okay.
Maybe 2 minutes, Katsu.
Okay, thank you. So the one technique we are now trying to do the surface enhanced Raman scattering, so it can enhance Raman effect to the force to the 6 on the surface of metal. So we introduced this metal into the cell, and we detect alkyne tagged drug with high efficiency. So we use this kind of GFGLMK, that's the name of the drug, and we tag this drug by alkyne. And so as you see, and we introduced the nanoparticles into the cell to enhance these alkyne signals. And by using this, we successfully detected this alkyne signal, and with enhanced very high sensitivity, with enhanced signal. And in this case, we just took about 6.5 seconds per frame. And with this time resolution, so we can actually take a movie of how the drug is introduced into the cell. So this is just a movie after introduction. But in our research, we tried to measure the dynamics of the drug. So that we started imaging before adding the drug, and during observation, we added drug and monitored how the drug is taken into the cells. And so this is the image. And 2 minutes after the imaging, we administered, not 2 minutes after. So we added the drug during observation, and as you see, the spots are showing the drug, which is green, increases with time. So this shows the speed of the drug uptake into the cells. And we tried a different concentration, 0 to 100 micromolar. And as you expect, so the speed and the amount of drug is taken into the cell increases with the concentration. So this is, I think, we believe, first example of observation of the drug uptake, which is not known before because it cannot be observed by ordinary microscope. Okay, so that's basically what I prepared today. So in our research, we started from label-free analytical imaging, but we applied Raman advantage of Raman scattering into this labeling and tag imaging technique. And we're now trying to improve the sensitivity by combination of the plasmonic and surface enhancement effect. Okay, so this is our collaborators. And, thank you very much for your attention.